Welcome to Scigest, your fortnightly science from the Bioeconomy Science Institute.
Autumn is a season of change. The mornings become colder, the days grow shorter, and forests burst into shades of gold, orange, and red. It's a transformation we've come to expect, but there's another tree that is turning red. Pine trees. Wait, what? You might be asking. Aren't pines supposed to stay green all year? Well, across New Zealand, some pine forests develop striking patches of red needles. At first glance, it can look alarming, as though the forest is dying. But behind those red needles is a fascinating story involving a microscopic pathogen, decades of research, and one of New Zealand's most important plantation species. Welcome to Scigest, brought to you by the Bioeconomy Science Institute, Maingi Tayo. I'm Darryl Herron, and today I'm joined by forest pathologist Dr. Emily McLay. Today we'll explore what needle cast is, why it turns pine forests red, how scientists uncovered its biology, and how that knowledge is helping us manage the disease. But before we learn more about red needle cast, Emily, let's start with you. How did you become a forest pathologist, and what exactly does a forest pathologist do?
Hi Darryl, thanks for having me on today. I actually started off at a lot smaller scale looking at crops when I studied university, so I was into kind of lettuce and things like that, and then got really interested in the interaction, I guess, between lettuce and other crops and the microbes that are associated with them. And from there I kind of went down the path of pathology and looking at the diseases of these crops. And then once I finished my PhD and did a little bit of work after that, I really scaled out and started to get interested in forests, which are kind of their entire ecosystems. And it's not only, the individual tree and the microbes associated, it's kind of this whole ecosystem that you're looking at. So basically a forest pathologist is someone who studies diseases of forests or of these trees, but really that kind of ecosystem and how everything's working together.
So the pines are turning red. Weird. In New Zealand, this color change is for different reasons, but this episode is focused on red needle cast. Now, what is red needle cast, and what is its importance, or what is its impact here in New Zealand?
So red needle cast is a foliar disease caused by a Phytophthora, so Phytophthora pluvialis, and it's also been associated with another Phytophthora, Canovii. The name is pretty fitting. It really describes what this disease is. So the needles of many conifers, so radiata pine is the main one. They form lesions which turn red, and when you've got enough of these needles in one place, you kind of have the whole foliage looking like this red hue. And then eventually these needles kind of become quite rigid and they really easily fall off the tree, so they're kind of being cast. And as you can imagine, these trees, which are normally, evergreen, suddenly become deciduous, so almost naked pine trees. And since they need those needles for photosynthesis and to grow, you can see that we can get a reduction in trunk growth. So radiata pine typically has kind of 3 years worth of needles at any one time. And so when you get an event like red needle cast, which makes all those needles fall off, you can actually see decreased growth for kind of 3 or even 4 years, while all those needles have to kind of regrow and you get a full crown again.
So that's going to have an impact on how long those trees are in the ground when you can harvest and the yields you'll be able to pull out that forest, right?
Yeah, absolutely. So it really depends on how many of these disease events you have as well. So sort of in really wet kind of areas of New Zealand, so kind of in the east coast of the North Island, sometimes these trees can get hit every 3 years. And so to kind of have the same growth as a tree, that may be sort of somewhere where it's not so disease prone, it's going to have to be in the ground a bit longer, or potentially you harvest at the same time and you end up with a smaller log.
So Phytophthora pluvialis. Difficult name for most people to say and even spell, even for scientists. Too many P's and too many H's. The most famous example our listeners may be familiar with is late blight of potatoes that caused the Irish famine in the 1840s. This was caused by Phytophthora infestans, which wasn't actually known at the time. So I've gone on a little bit side journey here. But plant pathogens themselves, disease-causing organisms, weren't even known to science back in the 1800s. There was speculation that this disease was caused by a fungus, but that was only confirmed in 1861, so nearly 20 years later, when Heinrich Anton de Bary looked at this pathogen under a microscope. And this was our origin story for plant pathology as a science. Back home, New Zealanders would be familiar with cold dieback caused by Phytophthora agathodicida, both examples of a Phytophthora, but different species. We now know today that Phytophthora are not fungi. So what are they?
So Phytophthora oomycetes, which, like you say, is a completely different kingdom from fungi. They're kind of commonly described as water molds and are actually kind of more closely related to brown algae than they are to fungi. Phytophthora is derived from kind of Greek, where it literally means plant destroyer, which is, like you say, really fitting because there are quite a lot of Phytophthora that are plant pathogens. And I think one of the reasons that they kind of are so adept at being plant pathogens is because they can produce such a wide range of different spores. So some that are specialised in survival, so kind of thick cell walls and can lay dormant for a while, and other spores under different conditions are highly motile, so they can swim through the soil to find their host. And so they adapt really quickly, really plastically to what the environment's doing, to how they respond and be able to cause disease.
Now, red needlecast has an interesting history in New Zealand because at that time, aerial Phytophthora, so you mentioned aerial, which means in the air, they weren't really common or well-known in forests. We'd seen some aerial Phytophthora causing diseases in horticulture and agriculture. Phytophthora ramorum, another species, it's a serious pathogen affecting different hosts, including forest species such as Douglas fir, larch, and chestnut. This was kind of emerging in the early 2000s, about the time that pluvialis would have been breaking onto the scene. And this was also the time, interestingly, when DNA sequencing was taking off to help identify organisms. Can you tell us a little bit more about the history of Phytophthora pluvialis, the causal agent of red needlecast here in New Zealand?
Yeah, so I wasn't actually around studying pluvialis at that point. I was probably still in my lettuce fields. But basically, Phytophthora pluvialis, we believe it comes from Oregon, where it doesn't actually kind of cause too much issues in its sort of native range. It then found its way to New Zealand, so we're not quite sure how many introduction events, but sort of based on the genomics, we think it's probably 1, potentially 2. And from there, it kind of expanded out in our forests, and it took a long time for us to actually realise what we had in our forests.
Yeah, and we didn't know what it was, right?
Yeah, exactly. So
we saw disease before it had a name?
Exactly. So people were seeing kind of these patches of red pop up in forests, and because aerial Phytophthora weren't really a thing in forests, we first started looking for fungi rather than Phytophthora, and we didn't find any fungi associated, and it wasn't until people started finding Phytophthora pinifolia or other Phytophthora overseas that we actually started to look in the needles for it. We have another disease in New Zealand of pine trees called PNB, which is another interesting one, and I've purposely not used its full name here because it was something that's been around for a long time. And again, initially we were looking for fungi because it causes the needles of pine trees to go red. Couldn't find any fungi, so we called it physiological needle blight. And then more recently, we found that older samples of PMB have been associated with Phytophthora canovii. So maybe that one is another example of a disease that, we kind of discounted Phytophthora because, we didn't think they were aerial things, but it actually might be the causal agent of that disease as well.
So you mentioned Oregon. This disease affects other species overseas as well, including western hemlock. Are there risks to other tree species here in New Zealand? And I'm thinking of potential alternative species the forestry industry might be looking to add to the state.
Yeah, so radiata pine in New Zealand is the main host for this disease, but it does affect other conifers as well. So Douglas fir would probably be the main one that the forestry industry kind of considers planting, where it does cause needlecast and it does cause cankers on the twigs as well. I think our other kind of alternative species like cypresses don't tend to be affected. Eucalyptus are unaffected by Phytophthora pluvialis, but there are other phytophthora that can infect those species as well, and even some of our natives. So totara, if we're looking at that as an alternative species, pluvialis doesn't infect it, but there is another phytophthora that can infect totara as well. So although pluvialis might not be coming for these other trees, there are other phytoptheras out there.
Knowledge is key when it comes to dealing with these disease outbreaks. It doesn't matter if you're a human or a plant. I'm thinking about COVID. When that arrived, we saw how decision makers were grappling with quarantine and movement restrictions almost daily as more information was coming to light about the virus and the many strains of that virus causing COVID. It was obviously a similar experience for the Ministry for Primary Industries and the Forest Owners Association when pluvialis was kind of being explored, when red needle cast was being explored. I mean, it didn't have a name back then. We didn't even have a pathogen name. There were obviously some concerns at that stage as well. So it infected needles, but the bigger question about market access. So could this thing survive in wood was a question. So how did the industry navigate that and management sort of early on with this pathogen to protect market access?
Yeah, so that was a lot of the really, really early research that was done on discovery of what was actually causing red needle cast in New Zealand, was looking at those survival spores that I talked about. So these are called oospores for Phytophthora pluvialis, and looking at whether they're capable of surviving on wood for, the duration and the conditions that a log may travel at. And it was found quite convincingly that the oospores were not viable after those conditions. So although it's causing problems in New Zealand, it wasn't seen as sort of a trade risk for export of logs, which was quite good for the forestry industry.
And eradication wasn't possible because we figured that the disease was quite widespread, right?
Like you say, it's kind of, we saw these patches of red pop up before we even kind of knew what it was. And so by the time we kind of figured it out, it was caused by Phytophthora pluvialis, and looked at where the disease was. Yeah, it didn't really follow the traditional kind of eradication protocol. Sort of moved straight into that long-term management role.
So now that we know more about red needle cast and the organism that causes it, you and other researchers at the Bioeconomy Science Institute, Maingitayo, with industry support, have been working on a risk tool for the disease. Can you tell us more about this tool and why it's needed?
Sure. So one of the main challenges with red needle cast is that it's historically been really unpredictable. We typically kind of have these boom and bust years where in certain years we'll see red needle cast kind of across the country, where in some places it's wiping out entire stands, and then the very next year we see none of it. And then on top of that, even within a given year, we know it's got these seasonal patterns where we see more of it in winter and less of it in summer, but we don't know where it's going to pop up, or kind of there's quite a lot of variation within the year as well. And so when a forester doesn't know if this disease is going to be problematic, where it's going to be, if it's even going to show up this year, it's really hard to manage or even to monitor. And so what this risk tool aims to do is give foresters a bit more ability to predict when this disease may occur. So it basically shows infection risk over time. So based on the climate, how likely is it that your tree or the needles may be infected on a given day. And so foresters can use this tool either at a regional or a forest level to look at the history of infection risk over time, as well as kind of future forecasts, and make decisions about whether they need to do something about the disease this year, whether it's a monitor, whether it's a apply control kind of situation.
And you've developed something on the side, I suppose, of this tool, and it's a red needle cast manual. What is in this manual, and where does somebody find it?
Great question. So we don't want to kind of just put this tool out into the universe and not give people kind of a bit of a guide on how to use it, or, we see risk is increasing, right? What do I do next? So this manual is kind of a bit of a one-stop shop on all the sort of applied R&C research that has happened to date, to try and give foresters and other interested people a bit of an information on kind of the disease cycle for red needle cast, the impact it may have on your tree, so whether you should apply control or not, different control methods, so either kind of one-off chemical control or looking at longer-term solutions, as well as running through that infection risk tool and how to interpret what the risk is doing in your estate.
And I'm quite curious in the development of this risk tool. We've talked a little bit about the importance of the life cycle and understanding the organism, and this tool is obviously based heavily on the disease itself and how that organism interacts in the environment. It took you some time to get this tool right, to get the data. What kind of data do you need to build a risk tool like this?
So there's a number of different types of models you can develop when you want to attempt to predict disease. The one that we wanted to go for was a process-based model. And so for a process-based model, you basically get that disease cycle. So you may have a needle that becomes infected. That infected needle has spores produced on it, and then those spores are able to move and infect another needle. So you get that disease cycle, and you break it down even more than that, and then you have to understand or quantify how fast each of those processes happen. And so we kind of went right back to basics and wanted to understand how climate would influence each of those different processes. So how does, say, rainfall and needle wetness influence infection, and how does temperature influence infection? And so we could kind of almost build a mathematical equation which represents that disease cycle. And so that took a lot of kind of really targeted lab-based studies, but then once you've done this work in the lab, you need to make sure it works in the field. So we kind of had an intermediate, which was trap plants, where we'd take pine trees and put them out where we knew there was disease for a set amount of time and look at conditions there. And then on the end of the scale, we have full-blown field trials where we'd monitor natural outbreaks over time and see how well our predictions lined up with what was happening in the forest.
And you studied various locations across the North Island to monitor red needle cast from season to season. How do you study a disease such as red needle cast on large trees?
With difficulty. So yeah, when you're dealing with something in that larger scale, so a tree itself is super tall. Sometimes you can't reach the foliage, sometimes you can't see the foliage. But then a stand of trees or a compartment of trees or an estate of a forest is bigger yet. So you kind of have to use all the tools at your disposal. And so remote sensing was something that was really important. Looking at individual trees would have things like trail cameras, which would take photos every hour of every day that we could then annotate and look at changes in disease over time. So that's for the really high frequency stuff. Or we even use satellite imagery to look at changes across years for quite large outbreaks. As well as remote sensing, there's a lot of molecular tools that you need to look at. So for this disease in particular, a lot of the action happens before you actually see symptoms. So infection and sporulation typically happen on needles that look green to the naked eye. So using molecular tools where we can sort of detect if it's infected or not before we actually wait for symptoms was really helpful as well. But yeah, you had to get kind of quite, creative with some of the solutions. So, if you go out there and there's a tree and the needle foliage is 15 metres high and you're like, oh, I really want a sample. In some cases, we'd get someone to come out with a shotgun and literally shoot the needles out of the tree so we could take them back to the lab and have a look at them. But yeah, the scale of things is pretty big.
You spoke of these booms and busts of disease. So there are periods where you had no disease. Do you know where the pathogen goes when it's not in the forest?
That is a great question, and one that we also want the answer to. So through some of these studies of understanding the disease cycle, one of the things we know is that pluvialis hates warm, dry weather. So typically we don't see a lot of it over summer, as well as, those kind of years in which the weather patterns are a little bit drier and windier. And it's kind of interesting, we talked about, aerial phytophthora versus other phytophthora earlier. Most phytophthora you're able to find in soil, and pluvialis is one that we really struggle with that. We've been able to detect pluvialis DNA in soil, but we really struggled to get live pluvialis out. So it could potentially be kind of persisting at sites in the soil or an alternative species, or in potentially pockets of slightly more favourable microclimate, but it is still a pretty big question mark of where it goes during those unfavourable conditions.
And this tool has been out since May of this year. Do you know if forest companies have begun to use this tool, and will you be measuring the impact that this tool has on management into the future, or their plans to do that?
So we've been working quite closely with a few forestry companies to try and get them to use it. So at the moment, we've kind of got a bit of a prototype tool on the FGR website. There's a regional level of this tool, which basically shows each of the region and then the history of risk as well as the current RNC risk. And then we've got more detailed versions of the tool as well. So in a couple of our case studies, we've got climate sensors within their forests. We've been working with them to sort of see when is risk rising, what are you seeing in the forest? Risk is falling, what are you seeing in the forest? So from these case studies, we're really trying to build in what is the best way to use this tool, and then if we can get a couple of success stories out of it, then we can kind of try and get it wider as well. So we do have a couple of workshops coming up kind of in some of these areas as well. So in August in the East Coast, we were working with a couple of forest companies there to talk through the manual and the tool, and also just make sure that what we're suggesting works well in sort of their organisational structure as well.
What is next for red needle cast management, for your research in trying to manage this disease better?
So for us at BSI, I think we're kind of in a bit of a phase where we've just completed a massive amount of work within the Resilient Forest program. And so now we're sort of taking stock a wee bit and kind of in the technology transfer stage. So there's a thing in science called the Valley of Death, basically, where you complete, a really good piece of science, but then how do you get it out in the real world? How do you get people using it? And so I think we're sort of at a stage now where we're taking a pause on doing too much further in the research space, and we actually want to start getting forestry companies to use this information. So like I mentioned, those workshops and the case studies, what we're trying to do is sort of go out, see what's actually working from the research we've done, where there's opportunities for potentially future improvement as well. And so from there, we'll kind of take a bit of stock, look at how well everything's working, and then move on to the next stage from there. There is more RNC research going on in the world though, so there is a bit of work looking at alternative control technology, so using antimicrobial peptides or other organisms, as well as work by RPBC looking at increasing resistance of trees to red needle cast as well.
So some breeding research going on with the Radiata Pine Breeding Company.
Yes, yeah.
Some additional tools people spray for red needle cast?
Yes, they do. Yeah. So one of the control methods that work is the use of cuprous oxide at relatively low doses. It's a preventative fungicide rather than a reactive one. So again, another challenge with red needle cast, that you have to apply it before you see those needles turn red. So that infection risk tool is really valuable for understanding when you should apply copper.
Before the disease arrives. So more to do with the industry now, the next phase, working with industry closer to put this tool into a space where it can be used, because the research, like I've said, has been decades. And Resilient Forest was, how long was that program?
I think it was 5 to 6 years. There was a bit of a COVID period in there where experiments started and then they suddenly had to be stopped. So there was, I think, a bit of an interim year in the middle.
So a huge quantum of work supported both by industry and obviously by the Bioeconomy Science Institute to develop this tool. It would be a shame if it was not used for the purpose that we see it, for managing red needle cast. And this can obviously be a template for other diseases, other foliar diseases of pine, correct? Because we mentioned that pinus species suffer from this red discoloration, but it's not always just red needle cast, right? There's other diseases as well.
Yeah, so disease models are kind of used in other sectors quite a lot. So horticulture has quite a few, but this red needle cast infection risk model is kind of the first for forestry in New Zealand. And I think it's quite exciting because there is an opportunity to follow this kind of pipeline that we've used for red needle cast and create models for other diseases as well. So Dothostroma is one that we've kind of potentially briefly mentioned. That's likely the most devastating disease in New Zealand for radiata pine. It's a little bit different in that we often spray after we see the red discoloration, but being able to kind of predict that disease a bit better would be really useful. And then another potential is kind of moving over into pine nurseries as terminal crook. where the causal organism for that, Coleotoxicum, there's a lot of models for horticultural crops for Coleotoxicums. There's a good base for us to kind of jump off if we wanted to develop disease models for terminal crook disease as well.
Wonderful. So you are paving the way for future research. It's wonderful to hear. Like the red needles falling from affected pine, we have reached the end of this Scigest podcast. Thank you, Emily, for joining us and sharing the story of red needle cast and the research supporting industry efforts to manage the disease. We look forward to catching up with you again in the future to see how your research progresses. And thank you all for listening. The next time you're walking out or driving through a pine forest in autumn, take a moment to look a little bit closer. Those splashes of red tell a story of fascinating interactions between trees, pathogens, and the scientists, like Emily, working to understand them. To learn more, you can visit our Scigest page for extra links and resources related to today's episode. You can also find many other interesting podcasts like this one, where we talk to the researchers about their amazing research and why it matters. And if you've enjoyed this episode, please consider subscribing through your favorite podcast app. And if you have a moment, we'd really appreciate a rating or review. It really helps others discover the podcast. Until next time, mā te wā.